Thank you for the introduction. Thank you all for coming. So today I'm going to be talking about sex-specific differences in colorectal cancer metabolism. A lot of the work I'll be presenting to you today has really just been acquired over the past couple of months by my very hardworking team in my lab. So it'll be really interesting to get your feedback on this as well. So colorectal cancer is the third most commonly diagnosed cancer in women and men in the US, and the incidence and mortality rate has been declining over the past 30 years, and that's due to improvements in colonoscopy screening and removal of precancerous polyps and earlier detection. However, if we look at the two sides of the colon, the right side of the colon, which is from the cecum, is there a pointer here? From the cecum up to the right hepatic flexure, and also the left side from the sigmoid up to the splenic flexure, we actually see a difference in outcomes for patients depending on where their primary tumor is located, where right-sided colorectal cancer patients actually have a poorer outcome. And this was highlighted in a recent meta-analysis where they looked at 1.5 million patients with colorectal cancer in JAMA Oncology a couple of years ago, and again they saw that right-sided colorectal cancer patients have a poorer outcome. If we have a quick look at the biology of the colon and the two sides of the colon, we do see some differences in the prevalence of molecular features. In the right side of the colon, we do see that the tumors are more likely to have BRAF mutations, more likely to have microsatellite instability, have a CpG island methylated phenotype, and also have gene expression patterns that are linked to higher metabolism and also immune infiltration with these cancer molecular subtypes 1 and 3 rather. But interestingly, what we also see is that women have a lower rate of colorectal cancer, but a higher prevalence of right-sided colorectal cancers. It's also seen that women develop colorectal cancer about 10 years later than men. And we believe there is a degree of protection from estradiol, which lowers their risk. But then after menopause, they again can form colorectal cancer. So there are some protective links to estradiol and colorectal cancer. We see that it can be linked to activation of estrogen receptor beta, which causes pro-apoptotic signaling, inhibition of inflammatory signals, and is also linked to modulation of cellular proliferation and the cell cycle. We see that in patients with right-sided colorectal cancer that ER beta expression is lower and also decreases with higher stage. Interestingly, estradiol is also linked to cholesterol and bile acid regulation, where estradiol downregulates cholesterol and bile acid metabolism, and it's thought that secondary bile acid levels would be much lower in the colon due to estradiol protection. Secondary bile acids are very interesting in the context of colorectal cancer because they are known to promote DNA damage and reactive oxygen species, and also modulate WNT and beta-catenin signaling and can contribute to colorectal cancer. And there have been some very high-profile papers recently which have also linked secondary bile acids to a lower immune response in liver cancers and liver metastases. So given that estradiol might be protective for women against developing colorectal cancer before menopause, and estradiol does have these links to estrogen receptor beta signaling and bile acid metabolism, and we hypothesize that after menopause, bile acid production increases and can cause a higher prevalence of right-sided colorectal cancers in women. So to go about this study, we have two main aims. First of all, we want to identify bile acid metabolites in tumor tissues from men and women with colorectal cancer. And then in concert with this, actually carry out a more systems level approach, looking at all the potential metabolites that might be present in colorectal cancers, and those are defined more in women with right-sided colorectal cancer, and those that could potentially affect tumor growth and therapeutic responsiveness. And we also want to link all of these metabolites to patient outcomes. So in order to measure metabolites, we use a tool called Metabolomics, and this is a systems level evaluation of all the metabolites that could be present within a sample. So when we talk about metabolites, we talk about those entities that are products of food ingestion and those that are synthesized de novo. They compromise classes such as amino acids and sugars and fatty acids, and they're very much regulated by upstream genome regulation of enzymatic reactions, but they're also regulated by the gut microbiome. It's also possible that very small perturbations to your local environment can affect metabolite levels, things like stress, things like lifestyle changes. If you have shift work patterns, if you have a change to diet, this can massively change your metabolome. And we can also measure another set of metabolites within samples that come from the environment. So these are chemicals that are not naturally produced in your body, but can enter due to things like drug use, cosmetic application, environmental pollutants, and food additives. And all of these different factors interact together to give each person an actual defining metabolic phenotype or a metabotype. And as you can imagine, this can be quite difficult if you're carrying out a study. But what it does do is help us understand disease heterogeneity and patient susceptibility to disease, and also variation in response to treatments. So this is just the setup of my mass spectrometer in my lab over at the School of Public Health. Here we have a liquid chromatography hyphenated to a high-resolution QTOF mass spectrometer. And this instrument actually allows us to do the systems level analysis, looking at all of the metabolites that could be present in our samples in a discovery-based approach. And this allows us to identify new metabolites and also new metabolic pathways that may be correlated with the disease. It's also possible to carry out a targeted, more hypothesis-driven method where we target, say, a handful or up to 100 metabolites of interest, and we're able to obtain quantitative information about these. And we're also able to target metabolites within tissues and do mass spectrometry imaging. So there we're able to understand a metabolite heterogeneity within a tissue. And the Dr. Vasiliu lab at School of Public Health recently acquired a mass spectrometry imaging instrument to enable us to do this. And this is just an example of one of the images that you can generate. So we have two different types of column chemistry, which allow us to analyze both the polar metabolomes. So these are water-soluble metabolites, things like amino acids and sugars. And then also reverse phase analysis, which allows us to look at the non-polar metabolomes, so things like fatty acids and triglycerides. So we're applying our metabolomic technologies to first identify bile acid metabolites in tumor tissues for men and women with colorectal cancer. So to enable us to do this, we obviously had to obtain some samples from patients. So my co-PI, Dr. Sajid Khan, who's a surgical oncologist at Yale School of Medicine, collaborated with Dr. Philip Patey at Memorial Sloan Kettering Cancer Center. And he acquired a very large tissue biobank from the 1990s, which had 760 colorectal cancer patients. So he collected primary tumors and also paired liver metastases. I think we have around 150 pairs of these tissues. Also mucosa, muscularis, and other types of metastases as well. All the samples are flash frozen immediately in liquid nitrogen and stored at -80. Basically from the 90s till now, they hadn't been touched. And this is a really valuable resource for metabolomic studies and also microbiome analysis, because they haven't been typically placed in FFPE, which we can't analyze using our instrumentation. So it's really valuable to obtain a resource such as this. So for AIM-1, we went very stringent at first, and we had some inclusion criteria for the patients who wanted to include in our study. So first of all, we took samples from patients where they just had right-sided colorectal cancer and left-sided. We didn't take any rectal samples or any transverse samples for this analysis. We also looked at just stage 1 to 3. As with stage 4 patients, they were most likely treated with chemotherapeutics before the samples were taken, which can affect our metabolic analysis. We also went for patients that were aged over 55 to first get rid of any effects of estradiol. So what we ended up with was about 220 samples from this cohort, where we had about 55 per group per sex and location in the colon. So I'm giving here a very poor diagram that I made of the liver and intestine, but this is just to show you how the bile acids that I quantified are formed. So we have primary bile acids, cholic acid and chenodeoxycholic acid are metabolized from cholesterol in the liver. They're conjugated to taurine and glycine, but primarily glycine in humans. They're then transported through the bile into the duodenum, where they then undergo hydrolysis by bile salt hydrolase in the ileum and colon to the primary bile acid again, cholic acid and chenodeoxycholic acid. And then they undergo another transformation by the microbiome, where they're dehydroxylated to the secondary bile acids. And DCA and LCA are known to be genotoxic. Some of these bile acids can be excreted in the stool, but the majority of them are taken up again for enterohepatic recirculation, and this can happen about 5 to 6 times. So when the DCA and LCA and CA and CDCA end up back in the liver, they get reconjugated again and then end up back again in the small intestine and the colon. So these were the different metabolites that we were targeting for our targeted analysis. And this is just some of the initial data where we have each of the bile acids here, the primary unconjugated and the secondary unconjugated, and then their taurine and glycine conjugates. And what we're comparing here is the fold change in bile acid levels when we're comparing left-sided tumors to right-sided tumors. And these are for all the stages combined and all women and men combined. And what we can see is a general increase in most of the bile acids in the right side of the colon, which we kind of expected. What we don't see is any difference to the taurine and glycine conjugated lipocholic acids. These are typically doubly conjugated in the liver to sulfates as well and excreted. However, when we start to look at across stage, we do start to see some interesting differences. And what we see at stage 1 is no changes to primary bile acids and secondary bile acids for the non-conjugated forms, but we do see higher levels of the taurine and glycine conjugated primary bile acids. And then when we move to stage 2, we start to see that the non-conjugated forms are increased and the taurine and glycine conjugated forms start to gradually decrease. And we don't see any differences at stage 3. So then we started to look a little bit closer at sex differences. And what you can see here is that actually there is a sex difference in terms of the bile acid localization in stage 1 and 2, where for women, there aren't any differences between left and right-sided for stage 1. And when we go to stage 2, we start again to see this trend of increased primary bile acids and slight decreases in the taurine and glycine conjugates for the men. So we have a working hypothesis that this could be potentially related to changes in the environment of the colon, which could be changing proliferation and utilization of the microbial enzymes. So at stage 1, it's possible that these two microbial processes are not happening. And at stage 2, we see this decreases in the taurine and glycine conjugates, and then this increase in the CA and CDCA and the DCA and LCA. So we have a working hypothesis that there could be differences in microbial activity at different stages in colorectal cancer. So we didn't see any differences in bile acids between men and women with right-sided colorectal cancer, but our hypothesis was that women before menopause would have lower levels of bile acids compared to after menopause. So we had a very small subset of early onset tumors. We had 8 samples from men with right-sided colon cancer and 10 from women with right-sided colon cancer. And so we just looked at the levels of the primary and secondary non-conjugated and their taurine and glycine conjugates. You can see here that there's no significant differences. And I hate to use the word trend, but you can see here that there are lower levels of the bile acids in the women with right-sided colorectal cancer, but we need to increase our sample number to see if this is an actual effect or not. So we believe that after menopause, the bile acid levels do increase in women with right-sided colorectal cancer. So just to summarize AIM-1, we see that both men and women have higher bile acid levels in the right side of the colon compared to the left. And there seems to be a difference in bile acid distribution when we go from stage 1 to 2. And we do see that the bile acid metabolic differences potentially are eliminated after age 55. So further assessments with the bile acid work that we want to do is actually to incorporate the clinical information. All this information is not in electronic form. So we now have somebody at Sloan Kettering pulling out things like BMI, hormone use, prior surgeries, racial ethnic groups, outcome and other medical history. And we would like to correlate the bile acid levels to patient outcome once we've assessed all these confounders. We also intend to do some receptor expression analysis and correlate with mass spectrometry imaging to assess correlation with metabolites and some histopathological features. And we're currently doing some microbiome analysis with Baylor College Core as well. So I'll move on to AIM-2, where we're doing our more systems level approach. I'm not going to go into this in detail, but this is just our workflow that we do in the lab. We've been really over the past couple of years optimizing a more population level analysis of samples for metabolomics, because you do get issues with batch effects and quality control. So this is something we've been optimizing in and developing code in R. Basically, after we obtain our data, we do a variety of multivariate and univariate analyses to find the most important metabolites and get rid of false positives. And then we carry out pathway analyses and metabolite identification, which is one of the hardest parts of untargeted metabolomics. And then we also validate using another platform and quantify the metabolites. So some of the differences that we assessed was metabolic differences between normal tissues and tumor tissues, differences between the right and the left sides of the colon, differences across stage, and also sex specificity. And there's about 30 different perturbations within this. So I'm just going to show a couple of the results. So the first couple of pathways that we've identified that seem to be really interesting is when we compare normal tissues to colorectal cancer tissues. And these are pathways related to arginine metabolism. So all the metabolites that are circled in orange are increased in men and women with colorectal cancer. Those that are circled in red are only increased in men, and those that are in purple are increased in women at stage 1 only. So this cycle here is the urea cycle, where arginine is transformed into the urea and also ornithine. Ornithine is transformed into citrulline and arginosuccinate. Ornithine is particularly interesting in colorectal cancer because it's decarboxylated into putrescine and enters the polyamine pathway. And ornithine decarboxylase is typically upregulated in colorectal cancer because it is also regulated by APC, which is mutated in colorectal cancer. But if we look at this other pathway, this is also potentially interesting because we see increases in this metabolite asymmetric dimethyl arginine. And this is formed after proteolysis of proteins and methylation of arginine residues. And ADMA has previously been seen in cardiovascular diseases because it's known to actually inhibit endothelial nitric oxide synthase and decrease nitric oxide production. Where this is particularly interesting in colorectal cancer is that it could potentially increase gut permeability, and ADMA has been associated before with E-cadherin. So it's possible that ADMA could increase gut permeability and increase microbial translocation as well. What we see is that in women, actually, ADMA is only increased in right-sided colorectal cancers, and we do see a slight increase in arginine in stage 1. And arginine is known to compete with ADMA for eNOS. In men, we do see an increase in citrulline, so it's possible that ADMA in men is metabolized into citrulline through this enzyme DDAH. and it's possible in women that DDAH is deficient. Interestingly, just as a side note, DDAH is also regulated by FXR, which is controlled by bile acid levels as well. So it's like a sort of overlap between all of the metabolites and these different pathways, which we're still trying to digest. Another pathway, this is the last one I'm going to mention, but which is also interesting in terms of women with colorectal cancer, is fatty acid metabolism. And what we see actually is that fatty acids are decreased in women with colorectal cancer, but we see an increase in the carnitine shuttle. So we're hypothesizing here that potentially women have increased transport of fatty acids by the carnitine shuttle and could be potentially used for beta oxidation and energy. So just to summarize our aim 2, we see multiple metabolic pathways that are altered in colorectal cancer that reveal stage and sex specificity. And we see, interestingly, metabolites that are related to NO regulation and also gut permeability. And we also see changes to energy metabolism in terms of fatty acid and the carnitine shuttle in women with right-sided colorectal cancer. So our future work is to investigate these metabolic pathways a lot more closely, as we've only really received this data over the past few months. And we want to use more of a multi-level approach by looking at gene expression changes as well, and also validating with any publicly available data that, to see if we see the same effects. We also want to look at links to cancer aggressiveness and patient outcomes, and then eventually go along the lines of therapeutic intervention. We're currently as well trying to validate this work by acquiring a number of samples from patients who are seen at Yale New Haven Hospital by collecting both tissue samples and stool samples. And potentially we'd like to move on to looking at blood-based markers as well. But we do see this systemic effect of increased bile acids in women with right-sided colorectal cancer, and we want to understand the relationship of all of these different factors within the different pathway changes that we're seeing. We're also looking to develop projects within endocrine disrupting chemicals in colorectal cancer and relationship to ER-beta, and also pharmacometabolomics to have predictive metabolites of patient response. So I would like to acknowledge all the members of my lab who have been working on the colorectal cancer projects. Yuping Kai and Nick Rattray, who since left to start a faculty position in Glasgow, Varia Mironova, Alvaro Santos Neto, and also Lingen Liu, who's helping collect the patient samples. My collaborator, Dr. Khan at School of Medicine, and also Dr. Chung at School of Medicine and Public Health, who's helping us with the outcome work. And then, of course, my collaborators at Sloan Kettering and the funding sources from Women's Health, Yale Cancer Center, and the NCI as well. And thank you for your attention.